Thank you for joining us for this evening's program on viruses and the immune system with Dr. James Baker. I'm Claire Noble, the Director of Programming for the Vail Symposium. On behalf of Chris Sable, our Executive Director, Dale Mosier, our Board Chairman, and the entire Vail Symposium Board, welcome. We are now in our 49th year of convening locally and thinking globally. Two items to be aware of before we get started. At the bottom of your screen, you'll see Q&A. That's where you'll type your questions for tonight's speaker. We'll get to those questions later in the program, and we'll get to as many as time permits. Tonight's program will run until 7 p.m. It's being recorded, and you'll be able to find that recording at vailsymposium.org. I'd like to take just a moment to thank some of the organizations and individuals who've helped make tonight's program possible. Our sponsors include the Town of Vail, Vail Resorts Epic Promise, the Antlers at Vail, and the Vail Daily. Our virtual programs are sponsored by Alpine Bank. Jeannie and Dale Mosier have underwritten the winter season, and tonight's program was underwritten by Dr. Keith and Mary Pat Rapp and Dr. Andrew and Lynn Kaufman. The Vail Symposium is also supported by a generous grant from the Frechette Family Foundation. Thank you to all of our donors. We have two remaining programs in our winter season that I'd like to just mention quickly. On Thursday, April 8th at 5 p.m., which is an hour earlier than we typically host our programs, we welcome Wes Moore, best-selling author and CEO of the largest anti-poverty organization in America. He'll be in conversation with Colorado Public Radio host Joanne Allen. And the final program of our winter season takes place on Sunday, April 11th, and features Roger Ferguson. He's the outgoing president of TIAA-CREF, and he'll be in conversation with Richard Bard, the founder and president of Bard Capital, and they'll be discussing the U.S. economic forecast. Now we turn our attention to a war waged at the molecular level between our body's immune system and the viruses seeking to invade it. Our distinguished guest is Dr. James Baker. Dr. Baker is the founding director of the Mary H. Weiser Food Allergy Center and the Ruth Dow Doan Professor at Michigan Medicine. A longer bio can be found at vailsymposium.org. And I've also provided a link to Dr. Baker's University of Michigan webpage, and I'll be putting that in the chat. Just a reminder to please use the Q&A at the bottom of your screen to submit questions for tonight's speaker. And now I will hand the program over to Dr. Baker. And Dr. Baker, just check back in with me when you are ready for questions from tonight's audience. We're just waiting for Dr. Baker to come on the screen, and he should be with us momentarily.
Claire, you muted me. Somebody muted me. There we go.
There you go.
Very good. Thank you, Claire. It's a distinct pleasure to be here tonight, and I have to say that this is a very remarkable group, and I'm so pleased to be able to talk to you about viruses and the immune system. A couple of things I want to talk about going forward. Initially, we were looking more generally, but I'm going to make this very COVID coronavirus focused because I think there's so much out there that needs to be discussed. It would be really helpful if we were able to sort of talk about this and clarify the issues about COVID in particular and how the infection happens and how the immune response protects us. And we now know that the immune response does protect us very well. The other thing I could say is that this is a very personal issue for me. I trained at Walter Reed. I won the draft lottery in 1971, so I trained at Walter Reed as a physician. I trained with Tony Fauci 40 years ago when he was 40 and I was 27. And my first trainee at Walter Reed was Debbie Birx. who now has gone on to different things. And Bob Redfield was a guy I've known since he was an intern and I was a medical student in the same service. I won't say that I give them the same recommendations I might have 40 years ago, but I do believe that this is a very personal thing. I believe that it hasn't been handled well on many levels. So I would really like to clarify for you many of the things that are going on so that we're all on the same page and we can all move forward and really get this pandemic ended, which I think will happen soon. So let me start my slides here. Claire, the way this is set up, I can no longer share my screen.
Okay, let me just check and
I
see
think-
what's going on there.
Yeah, you're the control right now. Against infection, it's your cellular immune response. It's the cells that go in and rid you of the virally infected cells, because that's the only way you eventually get rid of the virus. Okay, all right. So the one good thing too about cellular immunity is that it's very long-lived. And probably the closest correlate to COVID-19 is SARS. And SARS is made by a very similar virus that infects people. It doesn't infect people in a way that allows them to communicate it very effectively. It doesn't infect their upper airway, their nose, or their mouth. It only infects their lungs. So it's much harder to spread, and it was a much more contained pandemic. But in fact, SARS was present about 17 years ago. And in fact, studies have now shown that cellular immunity to SARS is still present to this day in people who were infected. So in fact, cellular immunity is our real hope for long-lasting protection against this virus. And it's very different in that regard from something like influenza, where we have to re-immunize people every year and everyone's at risk every year. And I'll explain why that is. And in fact, you're probably aware that people have shown that there's some cross-reactive cellular immunity from seasonal coronaviruses that cause colds. These are much different from the COVID-19 virus, but there's enough cross-reaction there that you get immunity. And in fact, antibodies do persist after the infection, particularly with the vaccines. And we're now seeing undiminished protection for 6 to 9 months after either the infection or the vaccine. So immunity is really our way out of this whole pandemic. So how did we develop these vaccines? And I think this is an important thing to see. This is probably the greatest success of biomedical research over the last 30 years. I was a little critical of calling this Operation Warp Speed because you don't really set your scientific investigation by naming it after science fiction that doesn't exist. And obviously the warp drive in Star Trek is something that doesn't exist and probably won't exist. But the idea was that this would be very fast vaccine development, and that actually was the case. There was actually a lot of vaccine. There were a lot of vaccines being developed before Warp Speed came to pass. And last spring, a little over a year ago, there were 170 vaccines that were in development. It was actually remarkable. But there were only a few vaccines, actually about 19, that had reached the point in human trials that showed that they might be useful. And those were the vaccines that were picked up and advanced by Operation Warp Speed. So Warp Speed was fortunate that they could sort of cherry pick things that people had already sort of done the preclinical work on and showed some efficacy. And what it then did was accelerate this. So in fact, they didn't have to accelerate the preclinical development. That was already done. What they basically had to advance were these few vaccines that were in clinical development. So Warp Speed accelerated and paid for 13 of the 19 lead formulations that were in clinical development. And they did some things that were very interesting. They had very specific viruses. The one thing they decided was they would not make vaccines the way we made them before. And you probably know that flu vaccine every year, we have to pick the right flu virus like 6 months or 9 months beforehand. We then take that virus, we adapt it to grow up in eggs. We then buy zillions of eggs and inject the virus into them. And then we have to purify that whole virus out of the eggs. And once we purified it, we inactivate it so it doesn't infect people. Well, the idea of making, literal metric tons of COVID virus did not appeal to the people at Warp Speed. So what they did was favor new technology where you didn't have to make virus, that you can actually take a component of the virus or the genetic material for the virus and try and use that as a means to vaccinate people. And none of those vaccines had ever been shown to work in humans. So basically they did what we will call the venture capitalist model, which is you accelerate and pay 13 companies with hopes that one will be successful and you'll get what you need. So in fact, they were shooting for maybe 1 out of 13. They set the bar very low, saying that all you had to do was show that you reduced infections by 50%. And the one thing they didn't do, they did a wonderful job of setting up and making these formulations. They really didn't address the logistics of storage, delivery, even getting enough needles to give the vaccine. And that's sort of why we had the hiccup at the start of this whole vaccine protocol, once we had the vaccines. So how did Warp Speed accelerate this? So the usual 5 or 10 years of clinical development before you get into humans, that all went by the wayside because they only picked candidates that were already shown to be safe in human beings. And so these clinical trials, what they did was give everyone all the money to do the clinical trials so that they could do them simultaneously. So once they had safety data, they could do the efficacy trials simultaneously. So rather than doing one for a year, reviewing the data, getting the FDA approval to do the next trial, they did all these trials simultaneously. And the other thing they did was they said, we don't know if your vaccine will work or not, but we're going to set up to manufacture it before we even know. So in fact, they started setting up to manufacture all of these vaccines, all 13 of them, before they knew that any of them would work. And of course, the only reason that worked was because, the government was paying for it. No company would agree to do that. Then they went in and rather than doing a full FDA review, they did a full safety and efficacy review. They did a mini review and gave what they call emergency use authorization. So in fact, rather than going through 15 years, I've got a couple of vaccines that I started developing 20 years ago. They're just now getting into clinical trial. And I've seen this from both sides. One of the things that they didn't tell you for disclosure's sake was one of my technologies was bought by Merck. And I spent a number of years as global head of vaccines at Merck. So I've seen this both from the academic side and from the actual corporate side. And no one would invest to manufacture a vaccine that you don't have data that shows that it's safe and effective. So what the government really did was make these processes sort of go in parallel, and that really reduced the amount of time it took to get the approval. They used the emergency use approval, so that was done quickly. And they had manufacturing up and running, so the minute they got the approval, they could start shipping vaccine. So it wasn't that they shortcut anything, They actually did all these, but they did them in parallel and they paid for them. So you didn't have to wait for one phase to end to begin the next one. And as I said, they only focused on new types of vaccines that didn't involve making the virus. Now, there is a whole virus vaccine that came out of China, and it does look like it might be effective. But there was no way that our folks wanted to deal with something that might cause, you could get a leak from a manufacturing facility and spread more virus around the country. So basically, the Pfizer and Moderna vaccines, which are remarkably effective, I'll talk about that in a minute, are RNA that codes for the vaccine. That's actually the genetic material for the vaccine in a lipid shell that helps get it into the cell, make this protein. That's taken up by your immune system, and you make T cells to eradicate the virally infected cells and B cells to make antibodies. So in fact, this is the cleanest and most defined vaccine we've ever made. There's no biologic component, there's no eggs, there's no virus. All you have in here is the recombinant protein made from the RNA, and it's really a very elegant thing. These vaccines were different. They were a different virus. Most all of them are adenovirus, which is a cold virus that was made defective, so it could not replicate. But as part of the genome in this virus, they inserted the RNA for the spike protein again. So it's the same protein that's in the Pfizer Moderna virus vaccines, but here it's being infected by a virus, and then the virus makes the spike protein in the cell. Your immune system recognizes that spike protein and makes cytotoxic cells and B cells to protect you. This is a little dirtier, and there have been problems with some of these vaccines, particularly the AstraZeneca vaccine, because it's actually a whole chimpanzee virus that doesn't replicate, but you're giving a fair amount of chimpanzee adenovirus to deliver the spike protein RNA to make it in the cell. So in fact, this is sort of an indirect way, whereas this is a more direct method for inducing this protein production, but both of them do work. And as I said, with the RNA vaccines, you couldn't just have regular purified RNA, because that's like a long string of spaghetti. You know, you can't get that into cells. It won't get inside. So what they do is contract the RNA inside of a lipid nanoparticle with charged materials. So in fact, the natively charged RNA binds to positively charged lipid particles, and it forms sort of an artificial virus that fuses with cells and delivers the RNA. But it's all synthetic. There's nothing in here that comes from any biological source. So remarkably, we were shooting for maybe 50% efficacy in the hope that we could protect against these this virus infection. It turned out to be the home run of all time. When I first heard the announcement of this efficacy, and I've worked in nanoparticle vaccines, so I believe in them, I was shocked by the results. But here are the results of the 3 vaccines that are now approved in the US and the AstraZeneca vaccine that's also gone through phase 3 and is approved in Europe. If you look at this data, it's remarkable because the blue line is the placebo group. And this is the start of the study. And you can see the blue line and the red line, and this is enlarged up here, parallel each other. So both the vaccinated people and the unvaccinated people in the placebo group continued to get infected till about day 14. And then after day 14, remarkably, the infections totally stop in the vaccinated group, where they continue at the same pace, undaunted in the placebo group. This literally stops infections in their tracks. And this was 110,000 people, half 2 in the active group versus every one in the placebo group. So in fact, this was a large group of people too. And the Moderna, which is, almost an identical vaccine, a couple of differences in the lipid component, gives almost identical results. The real key is that in both of these reports, the active arm blocks infection entirely. So in fact, these are over 94% effective. And we've now looked out at 6 months and they're 94% effective 6 months out. So you don't lose the immunity either. The one thing that you'll notice here is that the Pfizer is given twice at day 21 and the Moderna is given twice at day 28. And even though the infection stopped before that, the efficacy was actually not quite 100% until you got that second dose, because there were still some infections in these first 14 days. And we now know that if you don't get the second dose, both of these vaccines are only about 80% effective, and we aren't sure even how long they'll last. So it's really important to get both doses. The Johnson & Johnson vaccine is based on that adenovirus. You could see that it in the placebo group out until the end of this trial, which is about 91 days, you could see the infections still continue, and they continue in the active vaccination group, but at a much lower rate. So what you could see here is that even though the Johnson & Johnson vaccine is pretty good, it's about 80% effective, the infections continue. So it's not totally effective. And in fact, Johnson & Johnson is now doing a trial of 2 doses of this vaccine. My guess is if you get 2 doses of this, it'll be more like 90% effective. And in fact, the Russian vaccine that's very similar to this is 90+% effective after 2 doses. The real problem child here is AstraZeneca. And AstraZeneca, as you've probably heard, have had all kinds of problems with manufacturing. With their clinical trials, they made multiple mistakes. There have been questions about blood clotting and other things. And this vaccine, even with 2 doses, is not as good as the Johnson & Johnson. You can see here in the control groups, it never stops, infections never stop occurring. And they continue here. The immunity you get with this is not as effective. It looks like some of the variant viruses I'll talk about in a minute escape this. So in fact, of the 3 vaccines, I will label this opinion, but the Pfizer and Moderna are really the Cadillacs. They're the best vaccines. Johnson & Johnson is very acceptable. And the good thing is it blocks entirely from severe infections and death, even though it doesn't block from ongoing low-level infections. And the AstraZeneca is right now problematic. So I mentioned the idea of variants, and I'd like to talk a little bit about this because everybody's talking about variants right now, and I think it's confused a lot of people. And I don't think the news about variants is quite as bad as people make it sound. So what is a variant virus? Well, these are RNA viruses. And unlike DNA, which is a very stable thing, RNA breaks down and recombines all the time. So in fact, if you look at our genetic material, we're higher eukaryotes. Basically, it's very stable because it's double-stranded DNA and it's stabilized with proteins called histones. So it actually stays together. And if we have a mutation, it can result in cancer, but it doesn't change us as individuals. Even bacteria and things like yeast are very stable compared to RNA viruses. RNA viruses, break down all the time. Their RNA breaks down all the time, and it recombines in different ways, or you get substitutions. It's not very exact in what it does. So in fact, if you look at the size of the genome, RNA viruses like coronaviruses have very high mutation rates as compared to other types of things, and even DNA viruses. So it's not surprising that over time, RNA viruses change, and they change both for good and bad. And if they change in a way that the virus, can no longer replicate itself, then that virus, basically goes away, it dies out. But if they change in a way so that the virus is somehow better and can replicate faster or infect better, you get more of this, and the mutant virus, basically, which they call a variant virus, becomes predominant. So in fact, this is part of the normal way that viruses basically mutate. Now, the good news is that it's not like influenza. Influenza virus does similar things to what the coronaviruses do. They bind to cells and they have that molecular Velcro, and then on influenza, that's called hemagglutinin rather than spike protein. And hemagglutinin binds and internalizes the virus, the influenza virus. But the key with influenza is it binds to sugars, and sugars are on all proteins. So in fact, there are lots of options for this virus to bind to human cells. So in fact, this virus is what we call promiscuous, in that it will mate or bind to multiple different types of proteins. So that's a very important thing to remember. So any type of mutation in influenza, you still get binding to the cell. And in fact, that's why we have to have a new vaccine every year. We have to have an antibody that binds specifically to this when the virus is binding less specifically to the cell. So the virus can mutate and win and get around the antibody, but that's not the case in the coronavirus. And again, the key here is that this is binding to a protein, and it binds to a protein very specifically, a lot like antibodies bind to proteins. And as I said, you have this little finger up here that fits into that protein. And if you mutate this ACE2 binding region, the binding domain, too much, this won't bind at all. So in fact, you can't go too far away from the original virus and still have this virus binds to cells. So in fact, it's actually pretty much optimal at this point. And most of the mutations that can occur in this in the spike protein either don't improve the infectivity or diminish it in some way. The other thing is there's another part on here called the fusion protein. And this fusion protein is what actually inserts itself into the membrane of the cell and gets the virus into the cell more effectively. And there is more mutation in this area, and this is where some of the mutant viruses are. So they think it's not so much binding that we're worried about, is the fusion to the cell that we're worried about with the mutant viruses. But the good news is you can't change this virus as much as you can an influenza virus and still get a productive infection. So in fact, if you have an antibody, if you're making this RBD, which is basically what you're making in the vaccines that we now have coronavirus, and you make an antibody to that RBD, it usually binds to the RBD on the variant virus and still prevents this interaction. So in fact, there's now data that I'm going to show you that shows that our current vaccines were pretty good at this. So in fact, here's serum from people that were immunized with the Pfizer virus vaccine, the current Pfizer vaccine. This was one of the earlier mutants where you get an aspartic acid being mutated to glutamine at position 614 in the spike protein. And you could see that the immunity, the antibodies against this are totally, reactive with the new one. You could see with the UK variant that they're still pretty reactive. They're slightly less reactive, but in fact, there's more than enough antibody in here to neutralize it. And even with the South African variant that everyone's worried about, there's still enough antibody from the vaccine that protects against this. And in fact, yesterday in a study in South Africa, Pfizer showed for the first time that their standard vaccine protects against the variant virus infection. So in fact, all we have to do is get the whole world Pfizer and Moderna vaccine, and we got this thing licked, I think, and we've got most of the variants licked as well. But the really good news is that cellular immunity. We worry about the cellular immunity because that's the thing that really gets rid of the infection. And so we were worried that the cellular recognition sites in this protein might be mutated in the variants. But it turns out this is the whole coding sequence for this spike protein. So each of these letters represent a single amino acid. And you can see that there are orange letters here. And those are the mutants that have been described already. So there are mutants wherever these orange letters are. Here's a deletion mutant because there's a line through it. But the important thing is that the mutants in orange are different from these green sequences. And these green sequences are basically the cellular recognition domains. So not only do the antibodies cross-react, but all of the cellular recognition domains are maintained in the variants. So in fact, the immunity we get from these vaccines should protect us from all the foreseeable variants of this infection in the near future. This is a game changer. These vaccines will fix this. They'll end the pandemic. So the last thing I'm going to talk about, two last things. First, our vaccine adverse reactions. I am astounded that people are unwilling to get these vaccines. These vaccines are remarkably safe. And, as I said, they're synthetic, particularly the RNA vaccines. They have no biological components, no proteins, no toxins. The safety profile is remarkable and remains remarkable. There are only severe adverse events that we're seeing were fever and aches. And those are reported in a lot of people because you're making an immune response to the spike protein. And that shows that you're protected. And not surprisingly, they're more common with the second dose because in that second dose, you're developing a booster response. And they're also in people who've had the infection and get the vaccine, they're also more common because it's like a booster infection for them. No one has been shown to have died as a result of this vaccine. I mean, I have to say that because it's all over the internet. I can show you pictures of hundreds of people that were reported to die. In Norway, they were concerned that this was causing death. Well, it turned out that everyone died from this vaccine was in hospice care and weren't expected to live more than 2 weeks anyway. And they were giving them the vaccine, which didn't make much sense. But I could tell you, we have over 550,000 people just in the US who died from COVID, with over about 1,000 a day. And there's really, the risk-benefit ratio of these vaccines is remarkable. So the safety's there. One thing that I'm concerned about are allergic reactions, and I could tell you they were reported in the startup. There, unfortunately, were 2 of them the first day, but it turns out they occur in less than 1 in 100,000 doses. No one has died or been seriously injured from these reactions. They're 100 times less common than allergic reactions to penicillin. And there are multiple mechanisms here, and we're going to try and investigate this. And there are no uniform findings about what happens, but not a single person has had a life-threatening allergic reaction from these vaccines. And to tell you that we're taking this seriously, I'm now the head investigator on an NIH clinical trial that's 3,700 people at 30 sites, people with severe allergies who've been afraid or told not to get these vaccines. We're going to give them the vaccine under controlled circumstances and determine whether or not they react. And if they do react, we'll draw their blood, check their urine for what's causing the reaction. So in fact, if anyone has a situation where they've been allergic, they've been told not to get the vaccine, actually University of Colorado, our National Jewish is going to be a site for this trial. I encourage you to sign up and I can give you the sign up information after the talk. So where are we at right now to sum things up? This has been a remarkable pandemic. And in fact, the closest comparator is the Spanish flu, which was 1918 to 1990. We need to remember that that killed 50 million people. And it killed 50 million people in part because everybody who got the Spanish flu got sick. So in fact, remarkably, you knew who was infected, but even in people that got sick, some people in the same families would die and some people would recover. We need to learn a lot more about immunology to understand that, because it may be that some people just didn't develop a fast enough immune response. The reason we have 2.7 million people dying of COVID, which is much less than Spanish flu, but obviously worldwide much more than we'd ever hoped, is because we don't have an effective therapy for this now. If we had a therapy to treat these people while we were developing the vaccines, which is really what I thought would happen, that we'd have effective drugs, we wouldn't have nearly this many deaths. But we don't, and that's why we need to get everybody vaccinated. So what do I think is going to happen? First, I think we've got another 2 to 4 months of pandemic, and they'll be on again, off again, social restrictions and concerns while we get all the adults vaccinated. I believe the president, when he says anyone who wants to get vaccinated can get it by the end of May. And so all adults should be vaccinated by June, because it only takes a month. Students should be immunized for school in the fall. Yesterday, the results out of Pfizer showed that adolescents were 100% protected against this infection with the vaccine without any severe side effects. And what I think will drive this more than anything, the private sector is going to require vaccines. If you want to go to Disney World or a concert or a sporting event, you're going to need a vaccine. So in fact, I think that will get people to rethink this. The safety is certainly there, the efficacy is there. We might have ongoing small cluster outbreaks in areas where people who don't want to get vaccinated live. And really the biggest issue are people that refuse vaccination. If everybody gets vaccinated, this thing's over. The vaccines work. And the biggest concern for all of us is making sure the rest of the world gets vaccinated, because they serve as a reservoir to eventually develop very different coronaviruses that might come back and escape our vaccines. If we can get the whole world vaccinated, this problem is solved. And in fact, I put up here the remarkable resiliency of biology. This is the life expectancy in the US from 1900 to 1940. You can see here that the life expectancy in the US dropped 25 years in 1919 because so many young people were dying. But within 2 years, it was above the prior rate and kept climbing. So in fact, humans have dealt with pandemics forever. We recover on our own, but most importantly, we recover even faster this time because we have very good vaccines. So I'll end there. And what we can do is answer questions. So Claire?
Thank you, Dr. Baker. We do have a lot of questions and a lot of great questions. A couple of people have been asking the question around how long the vaccine is going to provide protection. And let me just read Dr. Lipnick's question. If SARS protected patients still have cell-mediated immunity, will COVID vaccinated patients need annual boosters, or will cell-mediated immunity provide immunity into perpetuity?
I actually think we're going to have long-lasting immunity from these vaccines. When you look at this, this is a very interesting virus. It infects a lot of people and doesn't cause any disease. That's why they can spread it, because they're asymptomatic. On the other hand, a little bit of immunity goes a long way with this. So looking at what we've been able to do with the vaccines, it's much better than the immunity we have generated to any other respiratory virus. And since the virus itself seems to act very much like SARS, I think there's a likelihood that we may not need another vaccine for 5 to 7 years. And at that point, it's only because there's been some very unusual variant that's come out, or because we haven't vaccinated everyone and suppressed this. We don't need any new smallpox vaccines now because we got rid of smallpox, mainly because of vaccines. So I think there's real hope that once we get the population immunized, we're going to be good to go for quite a period of time.
Well, Lauren has a question that has us circle back to the subject of T cell immunity. And his question asks, why do some people have a stronger T cell production than others?
Well, this is a great question. And you know, we really didn't know much about this coming into this. And this has been sadly a very interesting experiment of nature. They actually looked at young people that didn't have any predisposing conditions. You know, they went in and they looked at autopsy findings and they actually did genetics on these people. And what they found out is they actually have defects in the specific immune responses that protect you against RNA viruses. There are actually receptors in your immune system that cause you to react and make an immune response to single-stranded RNA, like in this virus. And these people had either defects in those receptors or defects in the signaling molecules in those receptors. And they wouldn't get ill, they looked pretty normal until they got infected with a virus like this, where you needed very strong and very immediate cellular immunity. So it turns out that there may be specific defects in these people that lead to the infection. Now that doesn't, when I say defect, I'm not saying that there's something wrong with them. They actually may have lower incidence of things like autoimmune disease because they don't have these signaling pathways. But obviously it's a deadly problem if they get infected with something like COVID.
We have a few questions around RNA technology. Both Stephen and Jeff have asked about this, the other applications of this really groundbreaking technology. Do you have any comments around that? For instance, Stephen mentioned that it may mean the end of seasonal flu.
Yeah, this technology will certainly be the new flu vaccine. And the reason for that is rather than trying to guess what the flu virus is going to be and grow it up in eggs and trying to adapt it, which takes almost a year, you know, in 6 weeks, you see what flu virus is actually already out there. You take the genetic sequence of it, you actually make the RNA and you put it into this vector and you've got the new vaccine. So one of the reasons we're doing the study about these allergic reactions is we want to make sure that we understand any possible problem with this technology, because you're exactly right. It's going to be used for probably almost all respiratory viruses at this point. The other thing that's very interesting is because it's so effective in inducing cellular immunity, the other thing you need cellular immunity for is to fight cancer. Just like killing the virally infected cells, cellular immunity kills the abnormal cells that are made by cancer. So in fact, there's some hope that this will be a platform to make vaccines against cancer. So I think this is a revolutionary technology. I think we got lucky because in fact, we immunize with RNA, and that's actually the thing that we recognize in the virus. So we actually kind of induce the exact right immune response that we needed for this virus. But I think it will be useful for other things, and it will be the new seasonal flu vaccine.
Buddy asks, why is the national media attempting to scare the public about the variants? And then also, why keep wearing masks if Moderna and Pfizer will finish off the pandemic?
Yeah, I think there are two things there. First, you know, the media, I think the media is not politically motivated. I don't think that that's the real driving force here. I think the driving force behind the headlines, the nightly news. I mean, I love Lester Holt, but my God, everything he presents now is breaking news, every single thing. And I think the whole thing here is to get people scared and get them to watch. I mean, they're going for ratings, and the way to get these ratings is to get people scared. And I think that's the bottom line with what's happened in the media. And after a while, they almost get addicted to it, you know, hyping everything. So I think they're hyping this now because the other problems are going away with the vaccines, and it's the only unknown they had left. But even now, this is being resolved, and the data from NIH and from other places in South Africa shows that these vaccines should be effective. And the second thing is why wear masks? I think we need to continue to wear masks until almost everybody is vaccinated. And the reason for that is that, Michigan. So Michigan, has been more restrictive probably than any state in the nation. We were in lockdown for almost the entire year. And then in February, they decided to things, ease the limitations because we had the vaccines. Well, there were two problems with that. Number one, not everybody had been vaccinated. And number two, we have the worst vaccine distribution system in the country. My wife had to go down to Toledo to get a vaccine. So I mean, it's not worked well. And of course, now what do we have? We have the largest increase in infections of any state. And that's because we have a totally naive population because we effectively kept them isolated. And then when they got out and started interacting, I mean, it was 30 degrees for a high here today. They aren't going outside and social distancing. So it was not surprising that this happened. I think until we get everybody vaccinated, which should be another 6 weeks or 2 months at most, I think we need to do that. After that, I think unless, there's something that none of us foresee, we need to declare this thing over and we need to declare our vaccine campaign a success and move on. And I think once we get everybody immunized, we can do that.
We have a few questions around the pediatric vaccinations. Specifically, how critical do you think this is, and how young can a child be to get the vaccination? And those questions come from Robert and Karen.
Yeah. I don't think pediatric vaccination is as critical as getting all the adults vaccinated. It's interesting now in Michigan and much like in Israel, where they did this initially, all the hospitalizations we're seeing aren't in the people over age 65 because they're all vaccinated. What we're seeing now is in the people from 40 to 65. They're the ones that are getting sick. So in fact, they need to get vaccinated. We need to get all these people covered and get them right down to 17. And once we get that whole population done, then I think it's appropriate and important to focus on children. They're much less likely to get sick. They're much less likely to have other types of problems, although there are some unique problems associated with this virus. But most importantly, they serve as a reservoir for this virus to remain in the population. And we want to get rid of that reservoir. We want to make sure that as these young people grow up, they don't lose immunity as they're growing. So in fact, I think it'll be important once they get all the adults immunized, to get the children immunized and get them immunized before they go to school next fall. I mean, I think we're going to have a normal school year next fall. And I think it's because all the adolescents who are the ones who are really at risk in the young group, younger children for some reason don't appear to be, but they're the first ones to get vaccinated. And we've got all the college and high school kids vaccinated. We're going to have normal, I'm going to be in Michigan Stadium next fall for football. We'll leave it at that.
So we've gotten several questions around the origin of the virus. The Biden administration just criticized the World Health Organization report. What are your thoughts on whether or not it naturally emerged or it was a lab leak, and does that matter?
So I can tell you a couple of things. First off, when you look at the mutations in the spike protein that lead it to be able to bind to the human ACE protein, they're nothing you could design with a supercomputer. These were things that occurred over time and eventually developed the ability to infect humans. And it's interesting, they've isolated this virus now from Vietnam, from parts of China, and other parts of Asia that are totally geographically distinct. And they've done it in bats, and they've done it in marmosets and monkeys, and it's there. And there are viruses that are almost identical to SARS-CoV-2. So in fact, it's in the wild. How did it emerge in Wuhan? That's something we don't know. I don't think it was purposeful. I certainly can't imagine anyone unleashing this on their own population. there have been a history of leaks at biodefense facilities, certainly in the Soviet Union with anthrax. My guess, though, is that this probably occurred at multiple sites. I don't think you could have a release from a single site, even if it was purposeful, that would yield the type of spread that we saw initially. My guess is that there were multiple little clusters of this infection in Wuhan very early on, probably 6 months before anybody knew about it. They weren't picked up as a global phenomenon. They were just thought to be little outbreaks of flu or something. And it wasn't until the numbers increased that they actually realized that this was the makings of a new pandemic. So I don't think the World Health Organization has gotten to the bottom of this. I doubt that this was a purposeful act, and I doubt that it was a single release from a single place. My guess is it was multiple places and it started percolating. You saw these little cluster outbreaks like we've seen in places like Houston and Miami happen in Wuhan, and that's what really triggered everything.
Dr. Baker, we are out of time. I mentioned to you earlier, I'd give you the last word. What last words would you like to share with us before we conclude tonight?
I can tell you that I'm incredibly pleased with the vaccines that we have and the speed that we use to get them. What I really hope is that everyone gets vaccinated and we make a concerted effort to get everyone in the world the best vaccines so that we can rid the entire human populace of this scourge.
Dr. Baker, thank you so much for your time tonight. Thanks to everyone at home for joining us. I hope you'll join us next week on April 8th. Remember that program starts an hour earlier at 5 o'clock, and we welcome Wes Moore and Joanne Allen of CPR. Thank you, everyone, and good night.